Just to begin with, I put my email address and our website information in the chat. So if there are things I say that you want to follow up on that we don't have time to address today, you will have obviously these charts to go back and look at. But by all means, feel free to follow up at any time. And if I can't answer the question, I'm sure I can find a colleague here at CASIS or on the NASA side that can work to address your question. So to begin with, I'm going to do a little bit of an introduction for who we are, because again, continuing in the prior theme, I'm a little bit out of my depth here. I am by formal training a lowly microbiologist. So thank you for bringing me into your fold today. I appreciate the kind offer of joining you today by Deidre, and express my gratitude to Hesse and to Connie especially for being so attentive and enabling me to join you today, and I appreciate your time. So we go to the next chart. What I wanted to do was just kick off an introduction from the global perspective and beyond about who we are and what we do. So once again, I work for CASIS. We are like Pharmacopoeia, a 501c3 not-for-profit organization. We are a mighty organization of about 55 people. And we support NASA in managing the International Space Station National Lab, which I'll introduce in a chart. But more globally, I just wanted to set the stage for part of what we as a species are doing in space. So I like to show this iconic photograph taken by Bill Anders, who was a member of the Apollo 8 crew. So this was taken on Christmas Eve, just prior to the Apollo 11 missions. And this Earthrise photo actually demonstrates, I think, more about what we learned from the Apollo program than actually putting humans on the face of the moon. So in the 1960s, the United States set forth on a mission to land a human on the moon before the end of the decade. But even before we reached that milestone in July of 1969, we had discovered something even more precious in the vastness of space, and that was how singularly isolated our planet was and how special it was. And that led, I think, and drove great social transformative change. It contributed to social transformative change across the world, but especially the United States in the 1970s and 80s, including a focus on toxicology, what we were doing not only to our environment, but to ourselves. Next chart. We've repeated this experiment as a species with the creation of the International Space Station. So beginning in 2000, the United States joined with the international partners in space to assemble an International Space Station operating in low Earth orbit, or LEO. That'll be one of the acronyms that I will undoubtedly use again in this talk. But the goal there was to create an environment, a living and working environment that could sustain humans for decades to come. Now, as we approach the end of operations for the International Space Station in 2031, a little over 5 years from now, we have discovered something even more precious than being able to maintain a continuous human presence in space for almost 25 years now. We've learned how space, and especially microgravity, can inform us about ourselves, our human bodies, our physiology, and inform us in ways and offer opportunities to develop new technologies and new discoveries that not only benefit humanity, but also help us to sustain our planet and to create new economic opportunities, new ecosystems off the Earth. Next chart. So specifically, the ISS National Laboratory, and forgive me, Connie, we call ourselves CASIS. Typically, when people see that, they do say CASIS, but I forget how they pronounce it differently. So it's an acronym. You can say it however you want to. But specifically, the Center for the Advancement of Science and Space is a not-for-profit organization that was selected by NASA in 2011 to manage the International Space Station National Laboratory. The ISS National Lab itself was created by the NASA Funding Act in 2005 to offer opportunities for access to space for other U.S. government agencies, for U.S. industry, and for academic institutions. Next chart. So specifically what we do is we support NASA in enabling access to the International Space Station for research off Earth for the Earth. So what's shown in this chart is just a breadth of the types of experiments and research focus areas that are supported on the International Space Station. On the left are many of those that are extremely important and are long-standing areas of research sponsored by NASA in partnership with other international partners and with other U.S. government agencies. And on the right are how some of those have grown into efforts that have some of their origins, at least within the creation of the ISS National Lab. So, for example, NASA has always had a very active program in science, technology, engineering, and math and workforce development engagement. But we've been able to expand that through NASA's interest in turning over the operation of low Earth orbit facilities to the commercial sector. And we're moving now into manufacturing and preclinical testing in the space environment where microgravity offers a significant advantage. Next chart. So very quickly, I wanted to go through over 50 years of microgravity research. So the thing I want to emphasize here to leave you with is simply that we have the capability now with the International Space Station and what comes after it to continue to leverage microgravity to benefit humanity. And that those benefits can occur at multiple scales. So currently operating on board the International Space Station is the Cold Atom Lab, which is offering new insights into Bose-Einstein condensates. Next chart. We've discovered through over 40 years now of crystallization research, the ability to manufacture crystals in the space environment, both for pharmaceutical applications and for non-pharmaceutical applications, foremost among those in the semiconductor industry, that offer improvements in crystalline form and structure that are far superior to any processes that we can currently use on Earth. Next chart. We've also now supported multiple companies with support and partnership of other U.S. government funding agencies and international partners who are looking at exploiting space for making diagnostic devices and biological products, bio-manufactured objects in space that also offer superior performance. Here, and the example that I've used here is from a company called Lambda Vision, which is looking at making artificial retinas in space that offer them significant advantages over the manufacturing processes they currently have. Next chart. I mentioned that this focus on crystalline structures has also supported new developments in semiconductors. That also extends to exotic optical fibers. So telecommunications is always looking for new improvements in devices that offer savings in transmission quality and performance. And Z-band are doped fibers. So we're moving beyond silicon glass, not only for semiconductors, but also for optical fibers. But some of those dopants cannot be reliably crystallized and manufactured here on Earth. So space offers the opportunity to do that. Next chart. And most importantly, I think for the audience here, we've been working with the National Science Foundation, National Institutes of Health, ARPA-H a little bit, as well as multiple arms within the Department of Defense, focused on the development of 3D models in space and looking at advantages in microgravity for manufacturing tissues that could be used either as models of human disease or in the future be used for clinical applications and treatment of human disease. Next chart. And of course, we continue to offer access to microgravity for whole organisms. Foremost among those, the most prominent feature of what we've done in low Earth orbit has featured crewed systems. So we have at NASA a rich database based on the human physiological responses to that environment. That's expanding rapidly with the advent of private astronaut missions, which offer far more diversity in age, gender, different ethnicities flying to the space environment with different purposes and different levels of exposure to the slightly elevated radiation environment in low Earth orbit, as well as that change in gravity and the effects it has upon human bodies. We also do fly model systems, including rodents, mice, and rats. I focused in this presentation on cell-based models because that's where we spent most of our effort, but we certainly have and support with NASA a very robust rodent research model in microgravity as well. Next chart. And next chart, please. I just wanted to give a little bit of a background on what's going on there, how microgravity works to our advantage and to our disadvantage. So please understand that microgravity, the absence of gravity, affects the physical properties of materials and mechanisms at a fundamental level, so that the transport of both energy and matter is affected in that environment. And these changes in the physical environment have very profound effects upon biological systems, whether they're at the scale of humans or all the way down to the scale of individual cells. So in the first chart up on the left, you see that in a 1G environment on Earth, you have buoyancy and sedimentation. So you have the separation of different density materials, or the things that are light and gas, like float up to the top in a liquid, and the heavier stuff settles out to the bottom. But in microgravity, there's more mixing that occurs. That has profound effects, not only, of course, on the physics in the system, but also on the biology of the system, so that we are able to achieve three-dimensional culture in the space environment without the needed introduction of bubbling or stirring or mixing or other things that we're spinning that we use here on Earth to create a neutrally buoyant environment. And the cells, it turns out, are sensitive to that. Buoyancy-driven convection. So if you need to transfer heat inside of a culture system or inside of a reaction vessel for non-clinical applications, for more industrial applications, you create these currents that are driven by buoyancy, these difference in densities, and as you introduce heat into the system. But you don't have that buoyancy-driven convection as the primary force in the microgravity environment. That leads to rather profound changes in our ability to look at different alloys for the manufacture of semiconductors, and also has impacts upon the manufacture of large monoclonal antibodies, for example. On the far right, the top is hydrostatic pressure. So this has an impact even in 384 well plate systems. On Earth, the deeper you go into the well, the more hydrostatic pressure there is in the system. From manufacturing viewpoint, this is obviously important to fermentation and carbonation processes that we use. In microgravity, there's an equal distribution of these exterior forces throughout the height of the liquid column that you're in. And at the bottom is one that most people don't typically pick up on, but containerless processing. So in two-dimensional cell culture, we do a lot to treat the surfaces for cell-based models so that they're not interacting with plastics in the system or other non-biologically compliant surfaces. In microgravity, you have the natural tendency of these cells or matter to be free-floating within the liquid environment. That offers rather profound advantages for cell culture, supporting tissue engineering and bioprinting applications, and is one of the major focus areas that we're seeking to develop for bioprinting and microgravity. Next chart. So the rest of the talk is just to touch on our portfolio and what we look at. So understand that everything I'm going to talk about with the cell-based assays are really derivative of what astronauts have experienced in microgravity for ever since we've had human-based exploration going all the way back to Yuri Gagarin's original flight. Some of these issues, these changes in physiological responses we have found mimic age-related physiological changes or disease states in human populations on Earth. These accelerated aging effects include accelerated bone loss, cardiac dysfunction, muscle wasting, immune dysfunction, neurological dysfunction. So we see, again, a lot of effects which we're able to mitigate in the case of humans in the space environment if they are going to stay in space for long periods of time. But only more recently have we learned that we can use those as variables in our application of disease models that actually offer us accelerated onset and progression of physiological conditions which mimic symptoms associated with human disease and aging here on Earth. Next chart. For example, astronauts returning from long-term missions aboard the International Space Station often present with visual changes. These were unknown about 10 years ago, but over the past 10 years, NASA and the international partners have discovered that visual changes were occurring in about half of the astronauts and cosmonauts who were traveling to space and staying for more than a few weeks at a time. There were also, for the first time, detected changes in the identification of injury, physical injury to the retina, and demonstration of increased pressure around the brain, which NASA has now described as spaceflight-associated neuroocular syndrome, or SANS. Next chart. So these rather profound effects upon vision reflect other profound effects of spaceflight on the human brain. So there's a global upward shift of the brain. This shift in body fluids in the body also impacts the brain and microgravity, resulting in a crowding of brain tissue and narrowing of the central sulcus. So there is overall about more than a 10% increase in ventricular volume, which is progressive with the longer you're in space, and of more concern for long-term habitation in a microgravity environment. The ventricular volume does not return to baseline even up to a year following spaceflight. Next chart. So again, these microgravity effects on the human body are well studied. There's quite a robust literature on the effects and how to mitigate those so that we can thrive in low Earth orbit, and how we have indications that we will be able to sustain missions to Moon and Mars. Keep in mind that a Mars mission is going to require the crew to be away from Earth for about 3 years, given current launch mission criteria. On the right, in graphs 2 and 3, are some of the more recent advances. So I mentioned that we have rodent research model for microgravity. We've certainly exploited the utilization of primarily mice, but some rat models as well in space, and have begun deploying many of the tools that you described here today down to single cell sequencing and transcriptomic profiles to understand what's happening at a molecular level in those avatars of humans as they are exposed to the microgravity environment. And then most recently, that's expanded into the use of tissue chips in space. I think of most interest to this group is focus on the development of kidney chips and liver chips for use in the microgravity environment. Next chart. So again, to exploit these cell-based models, we've taken advantage of the significant technological advances made in the use of stem cells. Next chart. And their application. So once you understand the utility of stem cells and how that we can induce pluripotent stem cells to be formed and have some insight into their ability to differentiate over time, offers insight into tissue engineering and regenerative medicine. So this is more on the application side of understanding how stem cells behave in three-dimensional culture, especially in the space environment. Next chart. And that, of course, has led to the significant investment in the global community in the development of tissue chips as disease models. So you've heard from some other speakers, prior, looking at the move away from animal models and the significant investment in beginning to adopt microphysiological systems as disease models to replace our reliance upon other animal models. They offer obviously significant advantages in that they are human. They offer the ability to move further away from generalized measures of different species, and in trying to translate those, transform those over to human experiences, but actually beginning to look at individual level variations so that we can have personalized chips that inform us about our specific responses to different sets of therapeutics given our projected risk of disease onset and progression as we age. These also, for the space environment, offer the ability to actually begin looking at higher throughput. We're limited with the rodent research model to only about 40 mice in a single flight experiment. We can do about 2 of those a year on the International Space Station. So we can get 80 mice up there a year is what that translates to, which is not enough to support even most preclinical investigations. But with tissue chips, we have the ability to automate that process and do much higher throughput. Next chart. So again, just to get back to the fundamental level, microgravity affects stem cell growth, differentiation, and function. And these effects recapitulate many, but not all of the effects observed during cellular senescence, aging, and the onset and progression of human disease. So again, what we have is another model, which, like tissue chips, is yet to be validated for clinical or even preclinical applications, but which offers many promising advantages to the current state of the art for understanding disease onset and progression. We're also, at the same time as we refine those technologies and learn more about stem cell behavior in the microgravity environment, beginning to look at manufacturing applications. Next chart. I like this quote. Chris Austin was the former director of the National Center for Advancing Translational Sciences, NCATS, at the National Institutes of Health, who defined, translation as the process of turning observations in the lab, clinic, and community into interventions that improve the health of individuals and the public. So with the advent of the ISS National Lab and this new pathway to space, we can add, in addition to the laboratory, clinic, and community, we can add space. So we are making observations on the International Space Station that we are beginning to see offer opportunity for interventions that improve the health of individuals here on Earth. Next chart. Few of these. So we've had an active partnership at CASIS with the National Science Foundation in multiple different areas. One of them, tissue engineering and mechanobiology. We've partnered with NSF now for 8 annual joint solicitations. And we've approached now $15 million in NSF funding, which have also included discoveries into the utilization of kidney and liver chips in the space environment as well. Next chart. We also have partnered with the National Institutes of Health, with a couple of the institutes and centers there, foremost among those NCATS, to offer opportunities for tissue chips and this accelerated onset and progression of physiologically manifested conditions which mimic symptoms in human patients in these chips. Next chart. So there's a lot more detail on the projects here. I just wanted to give an overview of what's going on there. So in 2016, we initiated here at ISS National Lab an Organ on Chip solicitation with about $1 million of funding. And then in 2017 and 2018, we joined with National Institutes of Health through the Common Fund to sponsor 9 multi-flight projects. That multi-flight's important, understanding that one of the limitations we have in getting to space is the expense of supporting research, but also the time involved. It's typically about a 3-year ramp-up period to get your science up to the International Space Station. So when we created this partnership with NIH, we wanted to offer investigators 2 flight opportunities as quickly as they were ready to fly, so they didn't have to get delayed by the cycle of apply for a grant, go through the review cycle, get funded, fly, analyze, publish, and then come back for your next grant, because that cycle is 5 to 7 years. And that's not working for anybody. Next chart. So tissue chips in Space One, please understand that tissue chips in Space One means that there is a follow-on, which is currently active. Next chart. So in the end, for tissue chips in Space One, the National Center for Advancing Translational Sciences and the National Institute of Biomedical Imaging and Bioengineering have committed well over $20 million. It's actually approaching $30 million in grant funding toward flight projects. We provided approximately $8 million for in-kind support, but more importantly, through NASA's contracts, the 50% of the resource allocation that we manage, that means that NASA's cargo contracts cover the cost of getting all of the science to space, having the astronauts activate, collect the data, and then get the samples and tissues and data all returned to the investigator. So our tax dollars have already paid for that part of it, so it's not passed on as part of the investigation to those other government agencies that we work with. The first go around in 2017 saw the award of 5 projects, followed on by 4 projects in the next year. Next chart. So the programmatic goals were to leverage organs and tissue chip technology that had been pioneered by investments not only by the National Institutes of Health and taken under the umbrella of NCATS, primarily at NIH, but also the investment that the Department of Defense had made, and also working with NIH and NCATS to develop multi-organ chips. Of course, a lot of that development led by Don Ingber's group, but also supporting others. Next chart. What we learned from those opportunities were that there were multiple other spin-outs from that that offered the significant opportunity to advance the technology in tissue chips. So in translating the benchtop experiment that investigators are doing in their own lab into something that could fit inside of a spacecraft and be operated by crew on the International Space Station, it led NIH into new directions in improving the validation of their tissue chip designs among the community. Next chart. Again, most important, I think, for the audience here in the first round of tissue chips was the focus on immunosenescence and the kidney model in the microgravity environment, but also support of lung host defense, which involved looking both at microbial interactions with therapeutics that targeted the tissues in the lung, as well as use of emulate chips to look at translation of drugs across the blood-brain barrier. Next chart. These are the investigators. Their publications, their peer-reviewed publications have just started to appear in the literature. I encourage you to follow up with those or reach out to me, and I'll point you towards their presentations and publications, which are currently available. Next chart. And we can, the change here, what I wanted to emphasize, that was with 1.1, we actually included some investigators that were seeking to improve their toxicological models. So these were people who were interested in using tissue chips to assess biomarkers, bioavailability, drug efficacy, and toxicity, using what were in this case single organ systems created on chips. But in round 2, which I'll talk about now, we're moving into the era of multi-organ chips. Next chart. These are the investigations and the institutions that are supporting round 2, or those that were selected in 2018. Next chart. And a list of the investigators whose publications are also now starting to appear in the peer-reviewed literature. Next chart. So to bring it back home to the ISS National Lab portfolio, this is how we built our portfolio. I invite you to visit our website, which is just issnationallab.org. We focused not only on these partnerships with other U.S. government funding agencies, we also partner with NASA in an area called in-space production applications. That's looking at the possibility of establishing manufacturing facilities off Earth that can create products, especially those in the biomedical realm, which offers superior performance in comparison to what we're able to do here on Earth. Of course, NASA continues to have interest and support research and continuing the human presence in space and moving beyond low Earth orbit to Moon, Mars, and beyond. And there's overlap there. So our portfolio focuses on those devices and those manufacturing means which can have benefit here on Earth, while NASA and Department of Defense, for example, invest in opportunities for those types of manufacturing applications which can benefit our continued human exploration of space. Next chart. So this is Tissue Chips in Space 2.0. This is a solicitation which was open up until about a month and a half ago, but we don't intend this to be the end of days for tissue chips in space. But tissue chips in space 2.0 built upon the results from the first go around started back in 2017 to begin looking at multi-organ tissue chip systems, and also hoping to hone the focus in use of devices for toxicity testing and personalized medicine. So the first round of proposals are currently in the hands of NIH to begin the review process. Next chart. I also wanted to emphasize we have within our own organization, Igniting Innovation National Lab Research Announcement, or NLRA. We do these Igniting Innovation solicitations in collaboration with our colleagues in the NASA Biological and Physical Sciences program. So collectively, we put about $7 million into the Igniting Innovation projects, the inaugural selection of projects just last year. We have another open solicitation this year in Igniting Innovation, which may be of interest to any investigators in this community. Next chart. And I have, I think, 2 more charts and then I'm done. So just wanted to emphasize again here that part of what we're doing with our portfolio in collaboration with NASA is seeking to expand the utility of 3D models, including organoids and tissue chips and microgravity, specifically in the area of targeting cancer. So we've been working as part of the NASA effort with other US government agencies for the Cancer Moonshot program. And what's listed here are just a few of the cancer cell types that have been explored in microgravity and have shown some accelerated response in that environment, which we believe can offer new opportunities for translational research in the future. Next chart. And the same with neurodegenerative models. So we've also learned that brain organoids specifically in the space environment offer opportunities for understanding the natural aging process that's in effects upon cognitive function, but can also offer insight into diseases here on Earth, which up until the present time have been recalcitrant to identifying effective therapeutics. Next chart. So that's the contact information for us. And if we could skip ahead one more chart and one more chart ahead, and then just hit the button, I think 4 times to see what happens. 1, 2, 3, 4, 5. Yeah. And hit it a couple more times. Hit it 3 more times. Let's see what happens. So I wanted to leave you with this. The International Space Station is slated to be deorbited in 2031, the beginning of 2031. But NASA and the nation are working on plans with commercial partners to build these new commercially operated platforms in space. So if you can hit the button, Connie, hit it 2 more times. Sorry, I forgot. I thought I had changed this one to all in one, but I didn't. Hit it 1 more time. 1 more time. 1 more, yeah. Perfect. So on the left is what we currently have. So we have the International Space Station operating at full mode, full operational mode. So we'll be continuing to support research on the station all the way through the end of 2030. And then beginning around the end of 2026, we'll begin to have new commercial laboratories operating in low Earth orbit that offer access to research, technology development, and manufacturing applications in space. And those will include both crewed and non-crewed free flyer platforms, which offer advantages for communities focused on different areas of access to microgravity. So with that, I thank you for your time and attention today. And if you have any questions, I'm sorry I went just a little bit long there, but if you have any questions you'd like to ask and you have time, please follow up. If not, I'd be more than willing to entertain them by email or phone. Thanks, Connie.
Yeah, no, really exciting presentation. I think it's worth having a couple minutes for questions. I think there's, this is a pretty new talk to this community, at least for me. So thinking of ways, one of the things when we chatted with Deidre and you is thinking of ways we could, work together or where there could be overlaps. So I'd love to hear that from the group. One thing to maybe kick off, a lot of really cool tools in them, the organ tissue chips, et cetera. What kind of outputs are available? Is it mostly visual or is there sequencing? Maybe you could speak to that.
Yeah, so it's all of the above. We are unfortunately still sort of in the regime of most of the analytical capabilities exist once you return the sample to the principal investigator's laboratory. So, but in space, right, the drivers of experimental design include mass, power, and volume to a degree that you're not accustomed to in a ground-based laboratory. So you do have, we have excellent imaging capability on orbit, but we've moved away. We have a confocal imaging microscope, but the difficulties that you have in maintaining sterility in a laboratory in your control are exacerbated in an international space station, which is at the end of the day a multi-user facility. So for example, there may be a rodent research experiment which is plugged into a wall, while on the ceiling is the microgravity glove box where you're doing cell culture, which you're using for cell culture one day, but the next day it may be supporting a ZBLAN experiment where they're pulling exotic glass fiber. So it's not a typical research environment where you have those distinct laboratories or facilities isolated. So in driving that, there are opportunities to do some analysis in real time. So certainly imaging data is collected, but we do a lot of that through on-chip imaging systems, which are, again, those are driving innovators for experimental design with increasing throughput, manufacturing applications, and increasing quality control in mind for terrestrial applications as well. So again, there's interest from the National Center for Advancing Translational Sciences and going down that path too. Things that will work in that space environment are also going to be more amenable to high throughput systems with clinical applications, especially for toxicological testing, where you have multiple compounds at multiple concentrations and you need to expose those either to multiple different tissue types or cell types. Those are drivers that are going in the same direction for us.
Thanks, Michael. Questions from the team? If there's any more, I guess one other question, since your background's microbiology, how much does that affect maybe the cell culture conditions compared to on Earth? And has that been looked at, like the maybe microbiome of the systems?
Yeah, so we've certainly, there's knowledge that the microbiome, increasing knowledge, right? We've just, I would say, started. The past 50 years of microbiology have been about moving away from just the things that we can cultivate in our laboratories into what all is there. And we've discovered, right, there are orders of magnitude more microbial diversity there than we were able to culture in our laboratory. And we were largely ignorant of the effects of that microbiome on our responses to disease or to therapeutics that treat that. So the technologies that have come online, metagenome sequencing, those are certainly have been employed on the International Space Station. It's an interesting ecosystem in that it is a semi-closed ecosystem in that there are frequent vehicles visiting, but certainly the microbial diversity on the International Space Station is depauperate compared to what we have on Earth. And it also is more human dominated. So think of it, in one way, it's similar to a hospital setting in that the microbes are human dominated. So you have a lot of human commensals and potential pathogens in that system. But it's also essentially like a locker room. It's not a clean room facility or a facility at all. But specifically from cell culture, most of it has been done from the point of view of what we could introduce into the system. There hasn't been a lot that looked at more naturalistic microbiomes that are introduced into these tissue chip systems. So it's a level of complexity that's coming in those models, but it hasn't. It's not certainly ready for prime time yet.
Sure. Let's go to Peter.
Hi, Michael. Great talk. So if I understood you correctly, one of the things that you touched on is this senescence gets either reversed or it stops progressing when astronauts and other organisms return to Earth. So what's sort of the time course on that? And are there explorations into mechanisms on how that works?
Yeah, so if you followed the twins study, right? So when Scott and his brother participated in a N of one study, right, looking at those responses, there was a lot of focus on telomere lengthening or shortening, right? We know that there were changes in telomere length, which prediction was that the telomeres in Scott, who was on orbit, would shorten more than those of his brother here on Earth. And the reverse was found. His telomeres actually lengthened over the nearly year that he was up there. So there's been data like that for quite a while now that indicated that there was some sort of accelerated aging effects at the cellular level, at the genomic level in space. But that obviously hasn't translated into increased risk or shortening of lifetimes for astronauts. There's a longitudinal study of astronaut health, which goes all the way back to the Mercury astronauts. And those individuals tend to be extremely long-lived and healthy. They haven't demonstrated increased incidence of cancer or other ill effects. But what we do know is there are profound changes in immune function, which has been traced back to the cellular level. There are profound changes in cellular activation and control systems. There's recent publications coming from Kat Jamieson at UC San Diego, specifically that have looked at markers that indicate pre-leukemic conditions in crew. And she sees if she were to detect the changes at the molecular level that she sees in crew samples that are returned or samples that are returned from organoids that are flown, she would put those patients into a bin of being at high risk for development of leukemia. But yet those astronauts, when they come back with samples, are not necessarily going to develop leukemia. So there are changes which we see which indicate that there's an accelerated aging process, but at some point the body is able to self-regulate and control that when the crew members return back to space. So it's at the level now where I would say there are very interesting indicators that indicate that those are obviously divergences from the model systems, these avatars, and what's happening in the whole organism. But they do offer interesting insights into ways that we can exploit microgravity exposure in that environment to get better insight into what are these key indicators of potentially adverse medical outcomes in humans in the future. That from the viewpoint of pharmaceutical development, those offer the ability to more rapidly screen what's promising and what's not promising from a drug development pipeline discovery. So there's some interest there. If we can increase the throughput to make it worth the time involved in getting there, that offers some interesting opportunities for increasing the rate of screening out compounds that are non-productive.
Right.
Thanks, Peter. Thank you so much. We'll go to Deidre and then we will take our break. So go ahead, Deidre.
Yeah, sorry. So I did want to say thank you for, the great presentation. And I do think that it's of, interest to the HESI eSTAR community, especially in the sense of the CIVM models and the microfluidic type models. Just as we heard this morning, looking at different, models and different mechanisms for carcinogenesis and how we can identify those gene signatures, say, earlier. And I think there's potentially a lot of advantage in this space in that if we can accelerate those processes, as we've heard earlier today, that it takes, cancer, a CARC study is about 2 years. If those, gene chips can accelerate that process and we can recapitulate that, say, in a microfluidic system, understanding what those, molecular initiating events or key events are, those things can be done and, validated. I know we're doing that work, but so much faster. We talked about potentially having to do additional work and it's just too long to do that. So, just thinking about something, like that those systems are getting currently developed and how they could sort of be utilized to identify biomarkers or predictive, assays, I think is really, a great way forward. And I think it's on its way. So just to mention that to, those folks online, to me moving forward and having some additional conversations, at HESI and the eSTAR committee and how we can utilize potentially and take advantage of some of the offerings that are available in, CASIS and others. I think there's some opportunities there.
Deidre, I appreciate your comments there, and I want to commend Deidre too. She's been a part of the ISS National Lab User Advisory Committee for several years now. Has played a very important role there. But in addition to Deidre's efforts, I realized from listening to you earlier today that we need more interaction with HESI. There's a lot, involved in the adverse outcomes pathway model that we need to begin thinking about how we can begin to deploy that on the International Space Station so that we can refine it, so that when these commercial platforms come online, they're ready to, hit the launch pad flying. I'll use that instead of hit the ground running when they move forward. So there's a lot here. We've begun to, have some meaningful, including NASA and other government agencies, are having very useful conversations with the FDA now, and thinking about ways to begin to move that model forward. But before we get to the serious regulatory talks, we need to understand where we could actually plug in and offer advantage to anything that you're doing now. And that requires conversation with the experts, where you're at, where your critical failure points are or needs for improvement that we can begin to test on station while we have it, so that they're fully formed and ready to go as these new platforms are designed and launched and begin to operate.
I will say one just quick, comment, and then I'll be done. But I would say, Michael, I think that eSTAR committee is one small committee, and I know that's what you're presenting to, and this was sent out to the other HESI committees, but there's a lot of different additional research that wasn't presented here today that I think could probably take advantage of this too.
Absolutely. Yeah.
Yep.
Yeah.
Yeah, great. Carol, maybe you want to make a similar point or another point?
Well, just one quick plug. I'm not sure if Vanita Chauhan is on the talk or not, but you're talking my language when you start talking about AOPs. And Vanita at Health Canada has been working with NASA and the Canadian Space Agency and internationally to develop adverse outcome pathways that describe they're aligned with the implications of space travel for adverse outcomes. And through these exercises, she's identifying the data gaps and the needs for methodologies and the biomarkers that we can use to measure biomarkers and methods that we can use to measure each of the events associated with different stressors like radiation and microgravity. And her AOPs end in cataracts, bone density loss, their neurological effects. So they're all there. And so if you're interested in hearing a little bit more about that, I can put you in touch with Vanita.
No, please do. And again, I wanted to say, I'm obviously deeply grateful for the service of the astronauts and cosmonauts and everybody that does that. The NASA Human Research Program has been working most closely with Canadian Space Agency, and their portfolio is, primarily and rightfully focused on maintaining crew health and performance. Our part of that is simply looking at what can we learn from the mitigations they identify and the conditions they're trying to address that can accelerate, the rate of translation here on Earth. And there's a lot of overlap there. So absolutely, anybody that you think would be willing to communicate with me, with us, with our organization, I'd love to learn more about those efforts and see how we can align what we're trying to accomplish, because it benefits all the communities, when we're able to do that.
Great. Thank you, Carol. Thank you, Deidre. Let's all give a huge round of applause to Michael. Michael, I think we'll be in touch. I think similar to what Deidre said, I think this is beyond just the genomics committee. It's really to a lot of HESI. I know our executive director caught the beginning of your talk, but I think follow up, with her and maybe board members would be really exciting. So let's plan to do that. And if anyone here wants to be involved in those conversations, let me know.
Yeah, thanks, everyone. We appreciate it.